and welcome to Immunity Time. Today we have here Professor Doctor Anja Lux from FAU-Erlangen, where she leads research on how antibodies work and how we can make them better. Her lab focuses on the fine-tuned mechanisms behind IgG antibodies, especially how Fc receptors and glycosylation shape immune responses. Using humanized mouse models, she explores new ways to improve treatments for autoimmunity, cancer, and even viral infections like COVID-19. She's also an award-winning teacher and a key part of FAU's growing immunology scenes. Anja, welcome, and let's dip into science.
Thanks for having me, guys.
Pleasure. And now, Ben, he brought today a very strange tea that comes in a powder, so can you explain?
When you go to Japan and you go to a sushi restaurant, they have this powdered stuff, and it's called Sencha.
Okay.
So I think it's made from the same thing as matcha, so it's got quite a grassy taste.
Okay.
But it's more subtle than matcha. I think matcha is quite bitter, so it's really refreshing. And when you go to a sushi restaurant, they have it on the table, and you can just make as much as you want. And they have like a hot water tap, so you can just
so they put the powder actually on the table like you do here.
Yeah, exactly.
Oh, cool.
Exactly. And then you can just make as much as you want.
But did you buy it in Japan? Or?
I did. I had to stock up.
I thought maybe you stole it from the restaurant or something. So Anja, could you tell us a bit about your path and how did you arrive here to Erlangen to be our professor?
Yeah. So I came to Erlangen more than 20 years ago now, which is crazy thinking about it. So after school, I spent 1 year in the United States. And from there, was thinking about what to do after. And I was always interested in like biomedical topics. And for whatever reason, it was clear that I wanted to be a researcher. Albeit I was less restricted on the topics. Funnily enough, I didn't like biology at all in school. So thinking then about it and looking at different options, I stumbled upon the molecular medicine program in Erlangen and immediately liked it because I thought this is exactly what I want to do in terms of being trained for research in the field of biomedicine. Luckily, after a bit of a waiting time, I got in. And then basically after my diploma thesis, which I did actually in the field of cancer biology, like looking at a molecule that's involved in colorectal cancer development, I had immunology classes as well. So I thought like, wow, this is interesting. And then I looked for PhD projects and I stumbled upon this new guy that recently came back from the United States and started his own lab. And that was FAU-Erlangen. So we had a brief conversation, and after that, I got stuck on the topic and dived real deep into antibodies, Fc receptors, and all that other stuff that I'm still working on now.
Yeah, we will have also podcast with Professor FAU-Erlangen, so he will also come here.
Yeah.
So yeah,
I was the first PhD student he hired when he came back.
And then it became a friendship. So why, like, it was Professor Nimmerjan, like, your mentor? Did you have any other mentor before that, like, kind of make your path into here? Like any professor that said something in a lecture that you said, oh, this is it?
I think before, not so much, to be honest. Looking back now at the professors we had at the time, all of them were more or less old white guys. So thinking back, I don't think they particularly encouraged me to follow this path. I think they provided feedback that was something that I could do, but not so much in terms of mentorship. But since then, obviously, there were a lot of colleagues, former and present colleagues, that give support that one can turn to for advice. And this is always really valuable. Even just like bouncing back ideas back and forth. This is something that is really important. And it's really, I don't think it's possible to follow this career path without mentors.
And also, like recently, we had kind of a bunch of chapters, all with male professors. Recently, we have posted the first one with a woman, with Andrea Tomacres, and now you are our second woman in the podcast.
Nice.
What do you
There
think?
you go.
Yeah, here we are.
Here we are.
So,
we're getting more and more.
Yeah. So what do you think about this? Like, did you have any problem while being a woman in science? Because we know it's a field that is not, I mean, it's becoming more and more female empowerment here, but like most of the names that we heard in the lectures are almost male.
Yeah. I think it's a slow progress. I mean, at the beginning, or for many, many years, it was just men. So, and positions become available one at a time. So it's supposed to be a slow process, but nevertheless, I think essentially the system is a system that for a long time, and maybe still does, discourage more women than men. I think this is also changing now, but not because the system is changing, but the men are changing. And they're also less willing to pay the price. And for the longest time, it was more the women that said, okay, I don't want to deal with this limited contract situation for such a long time, and the insecurity, the pressure, the feeling of not being good enough, I think is something that women feel more strongly than men do, at least on average. So it's really good that now there are more brave women that just try, and right now the situation is actually quite positive. So if you're a woman and if you want to follow this path, your chances are pretty good that you get a chance right now, right? Because the consortia need women, the funding agencies want to fund women as well. There's a certain pressure in society to do so. So it's becoming a bit easier, but on the downside, to be honest, this also generates a scenario where it's more likely that colleagues say, yeah, you just got there because you're a woman,
So
which is also not a really good
Yeah.
feeling, right?
Yeah.
As you can imagine. So it's a constant struggle.
So did you ever feel underestimated?
A story of someone saying something like that to you, like you only got here because of being a woman or something like that?
Yeah.
Really?
Sure, definitely. I mean, this happens.
Oh.
Yeah. I mean, this is not so much that they tell you to the face, but they say it in a room where you are about somebody else. Yeah. So I think finding the balance is really difficult, and I can understand that there's a lot of talented male colleagues that are less likely to get a chance nowadays because there is this pressure to promote women. So I do understand that. Doesn't mean I agree. But yes, I mean, the feeling of being underestimated is something definitely that happens, and not just to me. When you talk to other, especially women, this is something that routinely happens. You just have to deal and get used to it and deal with it. I think for me, sometimes I feel it even stronger because I've always been here.
Yeah.
And some of the older professors know me for a long time. I mean, some of them I even had in class when I was a student, like Professor Jeck, for example. Some know me from being a PhD student. So you grow up with this, and while this makes some things easier, being here for a very long time and continuously being able to work on projects also has these kind of disadvantages. So I think it's a mixture, to be honest.
Damn, it's really complicated.
It is, yeah. But I mean, it's a choice you make, right? And you do that on purpose, and you know full well when you enter the system what it's going to be like.
Yeah.
Yeah. And then, one just needs to try and see how far it goes. Yeah.
For sure.
I mean, you cannot succeed if you don't try.
Yeah.
That's a very good advice. So, okay, now let's go into topic. So let's talk about antibodies today. So we know that antibodies are produced by B cells, and we have IgGs, that is a type of antibody, but could you tell us a bit more?
Yeah, I think it's the most interesting type of antibody. Sure. So It's one of the main antibodies that we have in our blood, really at high concentration. It's also the most versatile. So it has the most spread of effector functions, basically. And with effector functions, we mean the stuff that the antibody can do once it has recognized its target. And the target can be like a bacterium or can be a viral particle, can also be a cell that we have in our body or a certain protein in our body, which would then have some less advantageous consequences. But so IgG antibodies bind their target really specifically. So they are optimized in a way to have really strong binding to very specific targets. They're also quite versatile in the type of target they recognize. So they can recognize like protein backbones, but also sugar residues, lipids. They don't care so much. As long as it fits like a key and a lock, that's fine. And when they do that, so that could suffice. So for example, a viral particle, strong binding of a lot of antibodies to that viral particle would already lead to protection, because then the virus can no longer infect its target cell in the body. So that would solve the problem. But sometimes you also need other types of effector functions, which would mean that the antibody recruits other components of the immune system, mostly the complement system, but also immune cells. And for the recruitment of the immune cells, we then need the Fc gamma receptors. So these are cellular receptors expressed on almost all kinds of immune cells that specifically interact with IgG and are then mostly responsible for mediating a lot of other different effector functions.
So that the antibodies don't just work themselves, they also work in conjunction with the cells in the immune system.
Yeah, basically they bridge kind of the adaptive and the innate immune system. Yeah, so we often talk about it the other way around, that you have an innate immune response. Later on, you get an adaptive immune response that leads to antibodies, but the antibodies mostly then need the innate immune cells to really perform their protective functions.
Okay.
Yeah? So it really depends on the situation, if the antibody can do the job on its own or whether it needs other components. But this is obviously the more interesting part for us as immunologists.
Yep.
Okay, and you have said Fc gamma receptors, but why the Fc receptors are so important? And what these Fc gamma receptors, what do they do? Like, because we heard that they can activate, but they can also inhibit. So how this really works? Like, first, what do they do? And then the rest.
This is a very fascinating family of proteins. So you have different types of Fc gamma receptors. The name implies or stems from the fact that they are receptors for the Fc part of an antibody, and especially for Fc gamma receptors, the Fc part of IgG. So the gamma is for the IgG. So you have also an Fc alpha receptor that binds to IgA, for instance. But we are focusing on the Fc gamma receptors, and there's multiple so-called activating receptors, which means if these receptors expressed on immune cells are bound by IgG, then this causes immune cell activation. And then there's one counterpart, which is the inhibitory. There's only one inhibitory Fc gamma receptor. If this is also kind of recruited into this island of receptors, then this will put a brake on the activation. Yeah? So it will induce intracellular signaling that limits the activatory signaling. So one really fascinating thing about Fc gamma receptors beyond their complexity is really how they work, right? So the one individual receptor has a really low affinity to IgG. So most of these receptors cannot on their own interact with a single IgG molecule. So what you indeed need is a so-called, what we call an immune complex. So you need IgG to be kind of in a clump, more or less. Be that due to binding to a bacterium, a larger protein, or a viral particle, whatever. So you have a lot of IgG molecule in very close proximity. And then you basically can bind the Fc gamma receptors, not just based on affinity, but also avidity. So a lot of weak interactions sum up to a sufficient interaction. And this also pulls the receptors together in the cell membrane. And this is needed for efficient cell activation. So we don't know yet how many receptors we need, and this is less an immunological question, maybe more a cell biology or even biophysical question that we would love to address in the future, to figure out how this process works and how this especially differs. And when you look at different immune cell types that express these receptors in different combinations and different numbers, so the system is extremely complex. We have 4 or 5 different receptors in different combinations, different numbers, and all of this with different affinities for IgG. And all of this kind of determines in a mixture the response. So the type of response and the strength of the response. And this is not known to the full extent yet, how this works for the cells.
So if you have more antibodies, is that always better?
Not necessarily. So the type of antibody matters very much. So you have a lot of different Fc gamma receptors, you also have different IgG. So you have 4 different subclasses of IgG, and not all of them work equally well. So when we are looking for an antibody that has a pro-inflammatory activity, it could, for example, provide protection in terms of a bacterial infection, you would rather want to go for IgG1 or IgG3 and not so much for IgG2 or IgG4. And this has to do with the effector function of these subclasses. So more of IgG4 might not help a lot, which is actually a really interesting thing because this was discovered in the context of the COVID-19 vaccinations, that there we have a really strong IgG4 response upon booster vaccinations. And this is also not understood what the consequences of this are. So just more doesn't do the job, so you need more of the right subclass. And right subclass kind of depends on the context or the biological question. And then also as a second determining factor, you would want to look at the glycosylation profile. So these 4 subclasses can come in 30 more different flavors. So different combinations of glycosylation or sugar residues attached to the antibody, which also affects what the antibody can do in terms of binding to Fc gamma receptors mostly.
And this glycosylation, it will matter for the therapy because there is a lot of therapy that we are adding antibodies, right? Will that matter at the end? Yeah,
that would definitely matter. And this is also already being explored. So for starters, the most obvious thing would be to cut off the glycosylation. So you can do this enzymatically or you can mutate the IgG, so that is no longer glycosylated. So nice thing about IgG that it's only glycosylated at one position. So that makes things very easy. So if you mutate this position and you thus produce a glycosylated IgG, this will no longer or hardly ever bind to the Fc gamma receptors anymore. So that is really clear. So if you are in a disease situation where you would want to use an antibody as a drug, but you do not want this antibody to bind to Fc gamma receptors, removing the glycosylation is a feasible approach. It's not 100% perfect, but it works pretty nicely. So this is already done. A bit more complicated is manipulating this glycosylation to fine-tune Fc gamma receptor binding. So this is now also done, mostly related to fucosylation. So it has been discovered that the attachment of fucose or the lack of fucose determines how well the antibody, the IgG antibody, binds to one distinct activating Fc gamma receptors. So there's now already drugs available that are produced in cell lines that do not attach this fucosylation. And these are supposed to be more active. Whether that is true remains to be seen. In preclinical studies and so on, and also in experimental work, it's quite clear. But whether this also really has a prominent beneficial effect in the therapy, I do not know yet. One would need to look at the studies.
Just to clarify, the ones you're interested in are IgG because those are the ones that you you produce in vaccination, or you want to produce, right? And the ones you use in therapies, like I think for arthritis, you can have intravenous immunoglobulins, right, as a treatment?
Yeah, but intravenous, so that's a bit different because the IVIG, the intravenous immunoglobulin, this is so far not produced recombinantly. This is taken from the sera of voluntary donors. So you need thousands of donors and many, many, many liters of blood. And then it's purified, filled in a bottle, and it can, first of all, be used as a replacement for people that don't have antibodies or sufficient amount of antibodies, but it can also be used as an anti-inflammatory treatment. And this is also based on glycosylation. At least that is what we think. So that it's the sialic acid as the sugar component, and IgG that is sialylated, so contains this sialic acid in its sugar moiety, is supposed to be more anti-inflammatory. So it has less binding to activatory Fc gamma receptors, supposed to be more working through the inhibitory Fc gamma receptors, so basically tunes down the immune system. In terms of cancer therapy, for instance, you would rather want an antibody that recruits Fc gamma receptors or binds to Fc gamma receptors more strongly, and there, like a low fucose version would be beneficial. So that is something to be looked at. And then is also the subclass that you can decide. So some therapeutic antibodies are IgG4, because for a long time we thought IgG4 is basically like Fc dead, which then suddenly became clear it's not. That's something, there's a consequence of 2 artificial in vitro experiments. So one has to also be careful. So it's a complex situation, so you have to really look at the context. What do you want your antibody to do? For example, where you address like rheumatoid arthritis, and therapeutic antibodies, IgG antibodies that are used for therapy of RA are, for example, TNF blocking antibodies. Yep. And they are supposed to not bind Fc gamma receptors, because you want the same thing as viral neutralization. So you want the TNF to be captured by the IgG, but no activation of immune cells on top. So you just lose it from the system, basically. Get rid of it. So you have to consider this very careful, what you're aiming for, but now we have the knowledge and the toolbox available to kind of manufacture a molecule that does what it's supposed to do. We still don't, beyond the fucosylation, we still don't really understand if there's any more chances of manipulating this. So we started to look into this in a very recent project that we just now started, trying to see if we can also preferentially target not just the Fc gamma receptor 3, which is responsible or involved in the fucose thing, but also other receptors, and if that would change anything in terms of, for example, viral protection. But this is something that we just started, and there's not a lot known along those lines.
So there's really, I guess, in the future a capability to customize these antibodies.
Yeah, that's the idea, possible. Right? And I think it's really striking that the stuff, not like the stuff that we directly do in the lab, but over all these years, over the last, 15 years, 20 years, the information that was gained in like basic research has started to be incorporated also in improving therapeutics. Yeah, so it started off with basically monoclonal antibody therapy, started off with mouse antibodies. And obviously, when you treat a patient with a mouse antibody, this will not work for quite a long time and will cause some severe unwanted side effects. So like improving these antibodies, making them more human was like the first major thing. To increase efficacy and also safety. And now we're basically at the next stage of engineering, and on a very more detailed level, basically. and figuring out what we can do to improve this. I mean, to be honest, these drugs are really expensive.
Yeah.
And we should make them as good as possible.
Yeah. And now that you're talking about that you are trying to make them better and everything, you have... you want more tea? Okay.
Thank you.
You can have
Should
more
also
tea.
drink some tea.
Okay.
Thanks.
Yeah, it tastes very good.
Nice.
Yeah.
I expected it not to like it, but I do like it. I don't know. It's just, okay, well,
This
whatever.
is really good.
Yeah, it is. So well, I come back to the question. You have humanized mouse models. I found that very interesting.
Yeah.
Can you develop that idea a bit for me?
So humanized mice is a very simple word for a very complicated model system. There's many different models all over the research community that people call humanized mice. So the model that we use is the humanization of the immune system by transplantation of mice with human hematopoietic stem cells. So we have mouse strains that hardly have any mouse immune system left. So they can accept a graft of human stem cells. And these human stem cells, we get those from umbilical cord blood. We purify them, and then we inject them into these immunodeficient mice. And from there, the hematopoietic stem cells basically have the potential to develop or to produce all the different types of human immune cells.
So it will be a mouse, but with a human immune system.
Yes.
And we expect them to react exactly the same as us?
That is the idea.
Okay.
Yeah, it's still not 100%. So one limitation is that the immune system is quite young. So it's not. It's the mice are 3 or 4 months old. So this is not what we see in those mice is not 100% of what we see in our adult immune system. But we do see all the major populations. What is for our research or for our purpose most important is that we have expression of the human Fc gamma receptors, and we have this expression exactly to the extent that we would see in a normal human or in a real human immune system. So we can actually use these mice to investigate human IgG activity. And we think that this works much better than doing this in normal wild-type mice.
So what's the advantage over the wild type? Oh wait, the normal lab mice compared to the humanized mice?
It's cheaper. So a major advantage is they are just available. You can breed them, right? So for the humanized mice, we have to generate each and every mouse. It for sure doesn't work every time, which can be a bit frustrating for the people doing it. So basically, tomorrow the student will go downstairs, inject the newborn mice with the stem cells, and we will know in 3 months from now, we will know if she has mice for experiments. So when you have a certain routine and you've done it quite a few times, then the chances are pretty good. So 70-80%. That the mice are overall humanized, but that doesn't mean that they're all the same. So that's a second thing, that they all look a bit different. So in terms of overall content of human cells, but also how many B cells are there, how many T cells, how many monocytes, do you see both monocyte populations, neutrophils. So each mouse is quite individual, which makes planning an experiment a bit more complicated. But on the good side, we have a situation where we can look at IgG, and we could show in the past that this is very useful to really predict activity, but also predict side effects, which is also quite important, especially when we look at like preclinical antibody development or just gaining a better understanding. It's not really feasible to, for example, look at the innate effector cells that are involved in mediating IgG activity in normal mice. Because the receptors are different. I mean, they're similar, but they're not the same. So you have different receptors at different combinations that have a different function. They all bind IgG, but beyond that, it's not that comparable. If you want to figure out which receptor is involved, or is it really rather the classical monocyte or the non-classical monocyte that is involved in mediating the protection or the activity, the IgG activity, then you're quite limited in doing this in normal mice. So this is really where the humanized mice come in. And now we want to also really explore this in terms of doing experimental models, IgG-driven models of autoimmunity, for example, and then using these humanized mice to check the efficacy of drug candidates. Because then we can really look at the impact of this potential drug on the human cells and not on mouse cells. So this is something that we are right now exploring.
Mm-hmm. Mm-hmm. Yeah, I mean, we talked to Professor Rosshart the other day about his wildling mice. I think you guys are sort of working along the similar lines of making the mouse models more translatable to human studies.
Completely different direction.
Yeah. Actually.
So we don't know. So based on what Professor Rosshart is doing, all of us might get into trouble, because the microbiome is certainly something that we ignore.
Mm-hmm. Yeah.
Which makes things even more complicated. I mean, on the good side, seeing the impact of different microbiomes on the humanized mice will be really interesting from a scientific standpoint. But the basic idea is the same, to get closer to what real life would mean. Mm-hmm. Yeah. Mm-hmm. So more heterogeneity than is represented in the normal mouse experiments. That for sure. But yeah, definitely different directions in doing so.
Even the fact that you have the same receptors that you would have in a human, that's already a big step.
Yeah, that's a major advantage for sure. Yeah, so if you do these type of experiments that we are doing and suggest to do, and you do this for you either do this for a mouse antibody in mouse, and then you can see like the normal interaction of the antibody with the Fc gamma receptors. But if it's a human IgG based drug candidate or antibody, we think that at some point it doesn't make any more sense to work on it in wild type mice, on black 6 mice. Mm-hmm. Yeah. Albeit one has to say that the normal mice have the advantage that you can just knock receptors out. Yep. And we are not that... I mean, theoretically, this is possible to do in the humanized mice as well, because we can genetically manipulate the hematopoietic stem cells. Mm-hmm. And we have done so in the past, but the question is, how efficient is that in terms of the complexity of the system? It's theoretically definitely possible, but we have not explored this to the max. So one really has to think about what are you looking for? Yeah, what type of information are you seeking, and then decide on what the best model is. And I think that the humanized mice are a very valuable piece of the puzzle, but they cannot be used to answer all the questions that are around in terms of investigating antibody activity.
Cool.
Yeah. And now, because we have talked before about autoimmune diseases like arthritis, and also I think you worked also on pemphigus, maybe, or yeah?
We have a current project running on pemphigus disease, yeah.
Yeah. So do you see different antibodies in those diseases, or they are kind of similar, or, yeah?
So both diseases in humans are driven by IgG autoantibodies. Mm-hmm. These autoantibodies are directed against different targets. So for pemphigus disease, it's a target in the skin. So we are working on a model of ABA, so epidermolysis bullosa acquisita, where the antigen is collagen 7 in the skin. We'll leave it at that. Whilst in RA, you would have antigens that are localized to the joint. Mm-hmm. Or at least, supposed to be localized to the joint. Beyond that, this is something we do not recapitulate right now in the research, because in the mouse models that we use artificial antibodies. Mm-hmm. We use either serum-derived or monoclonal antibodies to induce disease. So we are trying to, you gotta restrict your complexity to some extent.
Okay.
Yeah. So this is something that would be great to do. And this was in the plan to actually purify the potentially pathogenic antibodies from the serum of these patients and put this into the mice and then induce disease. So this is something we considered. Mm-hmm. For the pemphigoid disease, we let that idea go for now because it has become clear that the human antibodies from the patients would not bind to the collagen 7 in our mice because the mice don't have the human collagen 7, they have the mouse collagen 7.
Yeah.
So one could cross in the human collagen 7 as a transgene. Mm-hmm. So that would be an option. It's too many possibilities. You just have to decide which way to go. Mm-hmm. But yeah, so for the arthritis, I think it might be a bit easier. And we are now, thanks to the collaboration with the university clinics, we're in the process of obtaining serum from arthritis patients and trying to induce disease in the humanized mice with this serum.
So why didn't you have the same problem with the difference in the proteins in the arthritis compared to the pemphigus model?
So the colleague at the university clinics, she tested it, and she could show that it works. Okay. That the antibodies obtained from the serum of these patients bind to the joint. So either the target is more conserved or we're just lucky. Mm-hmm. But to be honest, this is something that we have to try systematically. Yeah? Might well be that it works better for some serum samples than for others, and it will take us a long time to figure that out and to kind of find or to define the key parameters. Mm-hmm. So going for like monoclonal antibody preparation is thus the easier approach to stabilize the system, and it also allows you again to investigate other things, right? If you would then ask the question, well, what's the difference between an autoantibody IgG1 versus IgG3? This is something that can hardly be studied with patient-derived samples. Whilst you can easily do that for monoclonal preparations. You can prepare mixtures of those antibodies, you can manipulate glycosylation, and see what the impact is. And then, try to kind of approach it from that direction. Yeah.
What do you think are the biggest obstacles in bringing the antibody therapy from the lab to the clinic?
I think ultimately it's the resources. Mm-hmm. So manpower and just money that is in the system. So I think once a candidate, so I looked it up particularly, so once a candidate is in clinical studies, chances are not too bad. Okay. Yeah, so somewhere between, I think, 1 to 2 in 10 actually get approved. Obviously, it's much less clear how many candidates fail before that.
Yeah.
And there's so many different reasons for that. And I think one key fact is which system you use to select and to test, right? We rely on in vivo models, so diseases are really, really complex. So if you want to find a drug or test a drug and investigate its safety and efficacy, you need an in vivo model system. But these always come with a downside. Yeah? Looking either on the target side or on the effector mechanism side, the cell type you're looking into, and so on and so forth. So that is, I think this is really something that can be quite frustrating I guess for the people that are actually like in this originally selecting part of the process. So you need to start with a lot of candidate proteins, antibodies, particularly in this context now, in order to ultimately have a candidate that works. And then it still needs to be better to something that was there beforehand. That's what makes them expensive, because you basically, for each approved drug, you have to pay for all the others that failed as well.
Yeah.
So this is certainly also a kind of drug. Monoclonal antibody therapies are, I guess, not really a drug that is feasible for the whole world's population. So this is something that is more prone to be prescribed here in the Western world, just because of the costs. And it's not just the costs of the drug, it's also the costs of delivery of the drug. Yeah, it's not a pill you can take, mostly. So I think this is something that severely impairs it. But nevertheless, the possibilities are endless. And that's why they are explored and further improved and changed and adapted to make them as good as can be, but also hopefully to get costs down at some point. I think that would be worthwhile to make that possible.
I mean, it takes a really long time to get one approved, right?
Yeah.
And at the end of the day, the longer it takes, the more money a company's going to have invested into it.
Yeah.
So they're going to charge more for it.
Yeah.
At the end of the day. Right.
Yeah.
Pharma
I think
companies.
easily 10 years, 15 years, easily. Just for like the clinical. I mean, we're not This stuff that we're either here we are working on antibodies that are already approved and we just have a closer look. So I was for quite some time looking into different types of B-cell depleting antibodies that are already approved in the clinics, and they work more or less well in different patients, but nobody really knows why that is the case. I mean, there are some ideas, but you can't really show in vivo, right? So that's where, again, the humanized mice came in, because this provided a model system where we could actually look at this in vivo, take out certain cell populations, try to block certain Fc gamma receptors, the complement system. You can't do this in humans, obviously. So a lot had been done in mice, but this didn't entirely fit to the human situation. So this is one option to really look at what is improved, take it from there, gain insights that ultimately maybe hopefully result in better drugs in the future. Or you get stuff that is really in a very early preclinical stage, and you try to figure out if it would work or not. Yeah. So it's basic research. Ultimately, it's still basic research.
And right now, the things that antibodies are used for would be like drugs that can block cytokines and, like you said, cancer depleting.
B-cell depletion.
Yep.
These are like the standard things,
Yep.
right? I think at least the ones that provide for the best income of the companies, like the cytokine blocking in autoimmune diseases, but also, the cancer cell specific, breast cancer cell, for instance, antibodies. But I think also now for quite a range of other types of diseases, like metabolic diseases and cardiovascular diseases as well. I mean, antibodies are really nice along those lines, right? I mean, you can generate them against any target, any type of, as long as it's found on the surface, you're good to go. You can block growth factor receptors. You can interfere with cell differentiation if you find the correct target, right? You can use them to deliver stuff. I think this is a really fascinating idea, to not use them as a cytotoxic drug themselves, but really just as a vehicle to get a toxin to the cancer cell, for instance, or in terms of like tumor vaccination, an idea that is being explored of delivering a tumor-specific protein to help train the immune system to respond to this antigen and develop an immune response against the cancer cells. So these are some nice features that can be explored for antibodies beyond the classical direct protective activity.
So there's so many diseases where antibodies could help.
Yeah.
Design antibodies.
Yeah.
I think it was also tried in COVID, maybe with SARS-CoV. something I might have read.
Well, like anti-inflammatory or blocking infection?
I mean, neutralizing antibodies. Yeah.
Yeah.
I mean, this is basically, these were developed really rapidly based on also the technical improvements and developments in the methods, right? So nowadays, this has been done by many researchers, companies, pharmaceutical companies also during the pandemic to generate antibodies that can directly be given to patients at risk. Yeah. So you don't wait for the patient to hopefully form an immune response and build its own antibodies against the virus, but rather you give the antibody directly. Yeah, so this was done. This is approved. And I think for the first candidates, they were actually really derived from patients. So nowadays it's possible, and I think this is the newest technique that is really promising in getting antibodies faster. Is you have your patient that has undergone an infection. You just take a few milliliters of blood, you sort the B cells, you sequence the B cell receptors, the heavy and the light chains, and then you produce the different antibodies recombinantly, figure out which of those, whatever, thousands, millions of antibodies is directed against your target of interest, and then you see if it's protective. So beforehand, you needed to immunize animals, mice mostly, and then do your hybridomas, and then humanize, and so that was a much slower process. But now with the direct single cell sequencing of the B cells, it's feasible to really speed this up, speed antibody discovery up. And I think this is also something that is explored for HIV. Where you also have few patients that do develop good antibodies, whilst vaccines are not able to do so, yet at least. So to do this in terms of discovery, this is a completely new world to speed up everything.
Right, so you can just take someone's antibody.
You can
or the sequence from the cells.
Yeah, you take the sequence from the cells and you produce this then in cell lines to generate your recombinant antibodies that you then use for screening. I mean, this was also, Thomas Winkler did it here in Erlangen, right? He was just, I think, a tiny little bit slower than colleagues in the United States. But essentially, they also did it here. Yeah.
So we could have like the next $1 million antibody in us right now.
The question is whether you will earn some money with it. I don't know how that is regulated, to be honest.
Will you sell your blood then? I don't think that's ethical.
I'll sell my antibody sequence.
You typically you're one of those voluntary donors.
Exactly.
But that would actually be a question to ask. Yeah, I never thought about that. Whether you have any rights to.
You get into Integrated Immunology Master, you have to donate your blood, and then we will sequence your antibodies.
I mean, it's a fast thing, right? So I think it's fascinating.
I totally agree. What do you think?
I think it's a bit scary.
Scary?
Why?
Well, it's like there's already so many issues in science about like bioethics and that sort of thing with like HeLa cells, like stuff like that.
You're not doing anything with the tissue anymore. You just sequence it.
That's
then
true.
It's gone.
But it's that information is part of my identity. It's like, I mean, if someone's taking a part of you and making money off it.
But you are saving people's lives then. Like we are getting in there.
Isn't it worth it?
I mean, it's not that someone can steal your bank account with your antibody sequencing,
If some company takes my antibody sequence and charges like, I don't know, like 500 euros a dose, when they could be charging like 100, and they're profiting off it in an unethical way, then I think I should have some say over how my antibody is used.
I would assume that you sign a piece of paper where you just give full rights, but it's certainly an interesting question.
That will be. We can make a poll in the podcast in the chapter. There is a poll. We will ask if you will do it or not, and then our listeners can give their opinion.
Maybe that's now why animals will never really get outcompeted completely. At least they will not sue you.
Exactly.
For any, also not typically, not really. Mice are not alive anymore afterwards anyway. So.
Okay, so as we reach the bottom of our cups, it's time for the last SIP question. That is 3 questions that we ask every guest that we have. They are a bit philosophical, but you can take your time to answer. So the first one is, if you could go back and speak to yourself on the first day in the lab, what would you say to yourself?
I think to celebrate the wins and the successes. There's so few of them. I mean, let's be real. And a lot of things just fail. A lot of ideas and hypotheses prove not to be correct. And we tend to go by the successes way too quickly. We're happy for a very brief time and then we just continue. But we should, I think it would be worthwhile to enjoy a bit more the stuff that works.
A week of holidays for success. At least.
It's interesting because Andrea, last time she said that you should be okay with frustration.
Yeah.
So you're sort of saying the flip side where you should be happy with the happy
and
things.
that is the other side of the coin, right? To not be too bummed or to learn faster, to not be too bummed. I'm not at all at the final stage where I can deal with something not going well. So very nicely. Yes, and it's funny that you mention Andrea because we recently had a situation where we were quite bumped, the two of us together. So that is the downside. But also, I think we spend way more time being frustrated than trying to be happy about the stuff that if something works, we should celebrate it. Yeah, I think that's. I'm not sure whether that helps.
I think
it's
that's
a nice
good life
idea.
advice. In general.
Yeah. I mean, at the end, these questions are also for us and for people that are starting in research to get like bit of hope or not only hope, but of, okay, so now we will start soon next year master thesis. So we also want to have advice.
You know this, right? I mean, you go into your studies, you prepare for months for an exam, you write it, then you pass it. You're like, yay, I passed it. Next.
Yeah, exactly.
Right? Or you fail, happens, then you're like bummed. Yeah, but you're bummed longer than you're happy if you pass it.
Yeah.
And you pass it well. And once it's always you just check it. Yeah, it's like a box you check because yay, I succeeded. What's the next? What do I do next? Instead of maybe one should just stop for a little while and really focus on it.
Yeah.
I try to. Maybe I should make a small note for myself and put it in
Yeah,
front of my
on the
desk.
computer, on the webcam next to it. Yeah. Okay, so question number 2. What's one mystery, scientific or personal, that you hope to see solved in your lifetime?
It's a very philosophical idea, but I have 2 daughters. And I know a lot of families with 2 children. And I always wonder how this is possible that the kids are so different. I mean, it's a really limited genetic pool, right? The circumstances are also really similar. It's the same set of parents growing up in the same house or apartment, whatever. Very similar circumstances. But nevertheless, you have the feeling that the kids couldn't be more different. Yeah. I would love to know how this is possible.
Yeah. Actually, that's very cool because I always, every time someone asks me about my family, my mom, there are 6 siblings and they are very different to each other. Yeah. Very different.
See?
And we don't know why.
That's isn't that strange?
Are you similar to your brothers?
I think we talk in the same way.
Okay.
Yeah, but in terms of how you think, how you
Very
act,
different.
personality, how you can you deal with success, frustration, and so on and so forth. It's so different. Strength and weakness is so different.
Yeah.
I mean, I find that really curious. This is from a genetic standpoint, I don't feel that this should be possible. But yeah.
Okay, and the last question is, if you could ask the immune system one question and get a clear, honest answer, what would you ask?
I think based on what we do in the lab, I would most likely ask why the immune system turns against us sometimes. I mean, why does it wreak havoc in some people and in others it doesn't? Why does it do so in young people? Yeah, I mean, this is more concerning even. There's so much we don't know yet. So if the immune system could help us understand, that would be really great.
So I think it was a very, very deep and nice conversation.
Yep.
Thank you very, very much.
Thank you.
I learned a lot about antibodies.
Yeah, for sure.
You're welcome. And again, thanks for having me.
Stay curious.
Stay critical.
And see you next time for some tea. It was very nice.
Reach us on Instagram at iimmunefau or send us questions, ideas, or feedback to iimmunefau@gmail.com. The equipment for this podcast is graciously provided by the Friedrich Alexander University Rundfunk Studio and Thomas Bauernschmidt.